In the United States, someone has a heart attack every 34 seconds. Heart disease is the leading cause of death for both men and women, responsible for almost 1 in every 4 deaths in the United States. One of the most well-known factors linked to risk of heart disease is cholesterol in the blood, and cholesterol comes in 2 forms. There's LDL, sometimes called bad cholesterol, and HDL, sometimes called good cholesterol. We know a lot about one of them. LDL has been studied extensively, and there's even a class of drugs called statins, which lowers LDL and lowers the risk of heart attack. So it's clear that intervening to lower LDL can have a significant impact on a person's health. But what about HDL?
For 30 years, researchers have been trying to understand the relationship between HDL cholesterol, the so-called good cholesterol, and risk for heart disease. What we know is that higher somebody's HDL cholesterol, the lower the risk of heart disease in the population. What we don't know is whether intervening to raise that HDL cholesterol will actually lower risk of heart disease. And this hypothesis has been difficult to test because we don't have drugs that specifically elevate HDL cholesterol.
But a new approach taken by Dr. Kathiresan and his colleagues offers an innovative way to put this to the test. Most of the time, when researchers want to test the effectiveness of a drug, they use a randomized controlled clinical trial. In these trials, patients are randomly assigned to either a treatment group, which receives the drug, or to a control group, which does not. Dr. Kathiresan's approach can't rely on a drug, so instead it relies on a trick of human genetics. Individuals typically carry 2 copies of each gene in the genome. Which copy a child will inherit is essentially a random decision, like flipping a coin. This phenomenon, sometimes called Mendelian randomization, offers a powerful means for testing connections between genes and disease.
This study highlights the value of human genetic information in understanding disease biology prior to developing and testing drugs in the clinic. What we found was quite surprising. We looked at 15 different variants that were associated with higher HDL cholesterol, and in each case, the individuals who carried the variant associated with higher HDL cholesterol were not protected from risk for heart disease. This tells us that drugs that might act through these mechanisms, the same mechanisms as the genes, probably will not affect risk for heart attack despite elevating HDL cholesterol.
It's important to note that there may be other genetic variants, ones that researchers haven't yet discovered, that do raise HDL and lower the risk of heart attack. Dr. Kathiresan and his colleagues are continuing to search for these causal variants.
We know that HDL is a great biomarker. It's quite useful in identifying individuals at higher risk of having a heart attack in the future. But we've shown that you cannot assume that raising HDL by any mechanism will help patients. Perhaps other mechanisms exist that can lower risk, but we'll need to keep searching for them.